Hello students, today we will be solving chapter 12 Respiration in Plants NCERT Exemplar questions. This is the disclaimer. So let us look at the learning objectives of our today's session. So we will explore and understand the fundamental concepts of chapter 12 Respiration in Plants by working through the NCERT Exemplar. This is the index. So from multiple choice questions to long answer type questions, we will be solving a total of 25 questions. So let us begin with the first question. The ultimate electron acceptor of respiration in an aerobic organism is? Options are option A cytochrome, option B oxygen, option C hydrogen, or option D glucose. So let us look at the answer. So the answer is B oxygen. So oxygen, it is the final or ultimate electron acceptor in aerobic respiration. What it does, it accepts electrons at the end of the electron transport chain. So once oxygen accepts the electrons, it combines with hydrogen ions to form water, that is H2O. Now all of this processes takes place in the inner membrane of mitochondria. So this is where the electron transport chain is located and where most of the cell's energy is produced. Now let's look at the next question. Phosphorylation of glucose during glycolysis is catalyzed by? The options are A. Phosphoglucomutase, B. Phosphoglucoisomerase, C. Hexokinase, or D. Phosphorylase. So let's look at the answer. The answer is option C. Hexokinase. So hexokinase, it is the key enzyme that starts glycolysis by phosphorylating the cell and beginning its breakdown for energy. So it catalyzes the conversion of glucose into glucose 6-phosphate by the use of ATP molecule in the reaction, that is phosphorylation. Now let us see why the other options are incorrect. So one option was phosphoglucomutase. Phosphoglucomutase, it is an enzyme. Its main job is to move a phosphate group from one carbon to another within the glucose molecule. And it is not involved in the initial phosphorylation of glucose, but rather it is involved in converting glucose 1-phosphate to glucose 6-phosphate, which is important in glycogen metabolism. Then if we look at phosphoglucoisomerase, it acts after the initial phosphorylation. So basically it changes glucose 6-phosphate into fructose 6-phosphate, which is a necessary step for glycolysis to continue. The other option is phosphorylase. Now this is an enzyme which catalyzes addition of phosphate group from inorganic phosphate to an acceptor. That is, it adds a phosphate group from inorganic phosphate, not from ATP, and it is mainly involved in breaking down storage molecules like glycogen or starch. Okay. Now let us look at the next question. Pyruvic acid, which is the key product of glycolysis, can have many metabolic fates. Under aerobic condition, it forms. Options are option A lactic acid, option B carbon dioxide and water, option C acetyl coenzyme A plus carbon dioxide, and option D ethanol plus carbon dioxide. So let us look at the answer. So the answer is option C, acetyl coenzyme A plus carbon dioxide. So pyruvate from glycolysis is oxidized, losing a carboxy group as carbon dioxide and forms acetyl coenzyme A. And this is the main pathway when oxygen is present. That is, this is happening under aerobic condition. And then this acetyl coenzyme A will then enter the citric acid cycle, where it is completely oxidized to carbon dioxide and water. Now let us see why other options are incorrect. So lactic acid, the lactic acid formation, it happens only when oxygen is not available. For example, in case of overworked muscles, and that is called anaerobic respiration. Ethanol and carbon dioxide, if we talk about these two products, they are produced by yeast during fermentation, which is another form of aerobic respiration. That is another form of anaerobic respiration. Carbon dioxide and water, they are the final products after complete breakdown of glucose in cellular respiration. And they are released at the end of the process, not immediately after glycolysis. Now let us look at the next question. The electron transport system, that is ETS, is located in mitochondrial. Options are A. Outer membrane, B. Intermembrane space, C. Inner membrane, or D. Matrix. So let us look at the answer. So the answer is C. Inner membrane. So the electron transport system is located in the inner mitochondrial membrane. Now this inner membrane, it is highly folded into cristae, which increases the surface area for electron transport system components. Finally, the ETS, it consists of complexes 1 to 4 and ATP synthase. That is, there are a total of 5 complexes, and all of these are embedded in the inner mitochondrial membrane. Students, now let us look at the next question. Which of the following exhibits the highest rate of respiration? The options are option A, growing shoot apex, option B, germinating seed, option C, root tip, or option D, leaf bud. So let us look at the answer. So the answer is option B, germinating seed. Now, germinating seeds, they have the highest rate of respiration. This is because they need a lot of energy to grow rapidly. So as soon as the seed absorbs water, the hydrolytic enzymes, they become active and they start breaking down the stored food. That is, they mobilize the reserve food materials, which is the stored food. Now, because of this, the seeds show high metabolic activity and quickly develop into tiny plants. That is, they germinate into a tiny plant. Now, all these activities, that is breaking down food, cell division, growth, all of these processes require energy. And this energy comes from the increased rate of respiration that happens during germination. So now let's look at the next question. Mitochondria are called powerhouses of the cell. Which of the following observations support this statement? Let us look at the options. Option A, mitochondria synthesize ATP. Option B, mitochondria have a double membrane. Option C, the enzymes of Krebs cycle and cytochromes are found in mitochondria. And option D, mitochondria are found in almost all plants and animal cells. So we have to tell why is mitochondria the powerhouse of the cell. So let's look at the answer. So this is because mitochondria synthesize ATP. So the correct option is option A. Now we know ATP or adenosine triphosphate, it is the primary energy currency of cells. And mitochondria produces most of the cell's ATP through the process of cellular respiration. So this connects the function of mitochondria directly to energy production. Now the term powerhouse is used because of this energy production, not because of how mitochondria looks or where they are found. So it's all about the function. Let's see the next question. The end product of oxidative phosphorylation is, the options are option A NADH, option B oxygen, option C ADP, or option D ATP plus water, that is H2O. So let's see what are the end products of oxidative phosphorylation. So the answer is option D, that is ATP plus water. So we know that complete oxidation of a glucose molecule, it produces around 38 ATP molecules, carbon dioxide and water. And this is all possible because of the energy released during oxidation of the reduced coenzymes. So this entire process, it is called oxidative phosphorylation. Now let us look at the next question. So match the following columns. Column 1 has molecular oxygen, electron acceptor, C pyruvate dehydrogenase, and D decarboxylation. You have to find the correct match for these options from column 2. We have alpha ketoglutaric acid, hydrogen acceptor, cytochrome C, and acetyl coenzyme A in column 2. And then you have to choose the correct option from the given options. So let us look at the answer. So the answer is option A. So first, molecular oxygen. This ultimately combines with hydrogen to form water at the end of electron transport chain. So this is the final step in aerobic respiration. So therefore, for option A, that is molecular oxygen, the correct answer is option 2, that is hydrogen acceptor. Now for option B, electron acceptor, so cytochrome C is an electron acceptor in electron transport system. So it is a mobile electron carrier present in the inner mitochondrial membrane. Now pyruvate dehydrogenase, it catalyzes the reaction that converts pyruvic acid into acetyl coenzyme A. And this step, it is the important link between glycolysis and Krebs cycle, which enables the cell to extract more energy from glucose. Now the next option for decarboxylation. So decarboxylation of isocitrate via or catalyzed by the enzyme isocitrate dehydrogenase, it forms alpha ketoglutaric acid in the decarboxylation reaction. So this step removes a carbon atom as carbon dioxide, helping the Krebs cycle progress. And therefore the answer is option A. Now let's look at the next question. Energy is released during oxidation of compounds in respiration. How is this energy stored and released as and when it is needed? So let us look at the solution. So first let's talk about the storage of energy. Now we have studied energy is primarily stored as ATP, which is adenosine triphosphate. So ADP combines with inorganic phosphates to produce ATP. So this process captures and stores energy in a new bond. So this high energy bonds between the phosphate groups in ATP, this is where the energy is actually stored. So breaking these bonds would release the stored energy. Now, some energy is also stored temporarily in molecules like FADH2 and NADH. So we have energy stored in ATP as well as temporarily in NADH and FADH2. Now, if we talk about the release of energy, so ATP releases the stored energy by breaking the phosphate bonds. This is a controlled process ensuring energy is released only when the cell needs it. So the equation is, or the reaction is, that ATP breaks down to give ADP plus inorganic phosphates plus energy is released. So this shows that when ATP loses a phosphate group, energy is released for the cell to use. Now this release of energy from ATP, it is well controlled by enzymes. So let's look at the next question. Explain the term energy currency and which substance acts as energy currency in plants and animals. Let's look at the solution. So a molecule that stores and transfers energy within living organisms is energy currency of the cell. This energy currency, it is used to power cellular processes as well as metabolic reactions. This means that every activity inside the cell, from building proteins to moving substances, will depend upon this molecule for energy. Now let us look at the main energy source. So ATP is the primary energy currency in both plants and animals. So ATP or adenosine triphosphate. It is used in immediate energy transfer processes. So cells don't store large amounts of ATP, they produce it as they go. Now let's look at secondary energy currency specifically in plants. So these are other molecules that help transfer energy in certain processes, especially during photosynthesis. So one example is NADPH or nicotinamide adenine dinucleotide phosphate. So this molecule is especially important in light reactions of photosynthesis, where it helps carry energy and electrons. So it is used specifically in photosynthesis, helping plants convert sunlight into chemical energy. Now let us look at the next question. Different substrates, they get oxidized during respiration. So how does respiratory quotient or RQ indicate which type of substrate, that is carbohydrate, fat or protein, is getting oxidized? So respiratory quotient RQ is equals to A by B. This is given. And we have to tell what does A and B stand for. And what type of substrates have an RQ of 1, less than 1 or more than 1. So let us look at the solution. So let's see first what is the RQ, respiratory quotient or RQ. It is defined as the ratio of carbon dioxide evolved to oxygen consumed during respiration. Okay. And RQ is given by A by B, which is given in the question, and this is equal to the volume of carbon dioxide evolved divided by the volume of oxygen consumed. Now let's talk about substrates. So substrates like carbohydrates, they have RQ of 1 during aerobic respiration. This means that the amount of carbon dioxide evolved or produced is exactly equal to the amount of oxygen used. Now, if we talk about proteins and fats, they have a RQ of less than 1, which means more oxygen is consumed as compared to the carbon dioxide that is being produced or evolved. And this situation often occurs during germination of seeds, when stored fats are being used for energy. And for substrates like organic acids, they have an RQ of more than 1. So this means that more carbon dioxide is produced than oxygen is consumed. Because organic acids, they already contain a lot of oxygen in their structure. And this situation occurs under aerobic conditions. Now let us look at the next question. F0, F1 particles, they participate in the synthesis of. Let's look at the solution. So F0, F1 particles, these are found in the inner mitochondrial membrane and they are involved in the synthesis of ATP, which is the energy currency of the cell. So here in the diagram we can see it shows diagrammatic presentation of ATP synthesis in mitochondria. So here when the proton gradient breaks, the energy released is used to drive the ATP synthesis process. This is the F0 component and this is the F1 component which drives ATP synthesis. So ATP is synthesized from ADP and inorganic phosphates. And this is present in the inner mitochondrial membrane. So let's look at the next question. When does anaerobic respiration occur in man and yeast? Let's look at the solution. So first let's see when anaerobic respiration occurs in man. So in case of humans, it occurs during intense physical exercise. So this happens specifically when the oxygen supply is insufficient. So in these situations, the muscle cells, they switch to anaerobic respiration. Why? Because the oxygen demand exceeds the supply. So a key result of this process is the formation of lactic acid in the muscles. So this lactic acid buildup is what actually causes the burning sensation or what we call as cramps that we feel after strenuous activity. Now let us look at the next part of the question, that is the case where anaerobic respiration occurs in yeast. So in case of yeast, anaerobic respiration, it occurs when oxygen is absent or it is limited. This process is known as fermentation. So yeast cells, they break down sugars without oxygen, which is important in making bread, beer and wine. So fermentation by yeast is used in brewing and baking. In baking, the carbon dioxide produced, it makes the bread rise, that is fluffy. And in brewing, the ethanol produced becomes alcohol in drinks. So the end products of anaerobic respiration in yeast are ethanol and carbon dioxide. So ethanol is alcohol and carbon dioxide forms bubbles in bread and beverages. So yeast, if we say, it is an important part in the food industry. So let's look at the next question. Which of the following will release more energy upon oxidation and arrange them in ascending order? So let's look at the options. Option A, 1 gram of fat. Option B, 1 gram of protein. Option C, 1 gram of glucose. Or option D, 0.5 grams of protein plus 0.5 grams of glucose. Let's see the answer. So 1 gram of glucose releases about 4 kilocalories of energy when oxidized. If we talk about a mixture of 0.5 grams of protein and 0.5 grams of glucose, this also releases about 4 kilocalories per gram. So this shows that combining these two doesn't necessarily increase the energy per gram. Then, if we talk about 1 gram of protein, it also releases about 4 kilocalories per gram. So even though proteins are mainly for building and repair, they can be used for energy when needed. And 1 gram of fat releases about 9 kilocalories per gram, which is much higher than glucose or protein. So this is why fats are the body's main long-term energy storage. So glucose and proteins, they yield the same energy per gram, that is 4 kilocalories. And fats, we saw that they yield the highest energy, that is 9 kilocalories per gram. So this makes fats the most energy-rich food molecules. And the mixture, it also yields 4 kilocalories per gram. And therefore, the ascending order will be C, D, B and then A. So C, D and B, they have approximately the same energy released upon oxidation, and A has the highest energy release upon oxidation. So let's look at the next question. The product of aerobic glycolysis in skeletal muscle and anaerobic fermentation in yeast are respectively and. So we have to talk about the products of aerobic glycolysis in skeletal muscle and then anaerobic fermentation in yeast. So let's look at the solution. So the product of aerobic glycolysis in skeletal muscles is pyruvic acid. So this is when oxygen is present. Now in anaerobic fermentation in yeast, the end products, they are ethanol and carbon dioxide. So here a different pathway is used by yeast when oxygen is absent. So here in the image we can see the major pathways of anaerobic respiration. So if we first talk about the incomplete oxidation of glucose during glycolysis, it forms pyruvic acid. And then this pyruvic acid is handled by different cells depending upon the requirement. So this pyruvic acid is converted to lactic acid in case of lactic acid fermentation. And this takes place in the absence of oxygen. And it is converted to ethanol plus carbon dioxide in the absence of oxygen in case of alcoholic fermentation. So here the end products are ethanol and carbon dioxide. And if we talk about skeletal muscles, the product of aerobic glycolysis is pyruvic acid. So let's look at the next question. So we will be beginning with short answer type questions. If a person is feeling dizzy, glucose or fruit juice is given immediately but not a cheese sandwich, which might have more energy. Explain. So let's look at the answer. So first let's talk about quick absorption. See, glucose and fruit juice, they have simple sugars and they are rapidly absorbed into the bloodstream through the digestive system, and therefore we see an immediate effect. So simple sugars, they will raise the blood glucose levels within minutes, providing a quicker relief from dizziness, which is why someone feels better so quickly after having juice or glucose. Now here, no digestion is required, unlike in case of a cheese sandwich, which contains complex carbohydrates, proteins and fats. So all of these will first have to be broken down before the body can use them. However, if you compare it to simple sugars, they need minimal digestive processing. So in case of emergencies, immediate energy is very important. So when someone is dizzy, immediate energy is important, and waiting for complex food digestion, it will only worsen the condition. Finally, liquids like juice, they are easy to consume when someone is dizzy compared to solid foods like sandwiches. So we see that in case of glucose or fruit juice, there is quicker absorption, there is immediate effect, minimal digestive processing is required, it is an emergency response, and the consumption is easy. Let's look at the next question. What is meant by the statement aerobic respiration is more efficient? So let's look at the solution. So aerobic respiration produces more ATP. It generates 38 ATP molecules per glucose molecule compared to just 2 ATP in case of anaerobic respiration. So in anaerobic respiration, 2 ATP molecules are coming from glycolysis. And this is the only energy that is produced in case of anaerobic respiration. If we talk about aerobic respiration, there is complete breakdown of glucose. That is, glucose is fully broken down into carbon dioxide and water, releasing maximum energy. So aerobic process, it is more energy efficient. Now the third thing is about toxic byproducts. Basically, aerobic respiration, it does not produce toxic substances. But anaerobic respiration, it can create various toxic substances like lactic acid or ethanol, which can be harmful if they build up. Next, aerobic respiration, it is a continuous process. It can keep going for long periods without causing fatigue because it doesn't produce harmful byproducts. Now, another important point is that aerobic respiration uses oxygen as the final electron acceptor. And this allows complete oxidation of glucose, ensuring that all the energy is extracted. Moreover, aerobic respiration, it provides energy to power complex organisms. So this high efficiency is what allows larger and more complex life forms like plants and animals to survive and thrive. So without aerobic respiration, life would be limited to only simple forms. And therefore, aerobic respiration is more energy efficient. So students, now let's look at the next question. Pyruvic acid is the end product of glycolysis. What are the three metabolic fates of pyruvic acid under aerobic and anaerobic conditions? And we have to write in the space that is provided in the diagram. So here you can see we have to fill in these three boxes. So let's look at the solution. So let us look at the three metabolic products which are formed under aerobic and anaerobic conditions. So the first is lactic acid. This is produced in animal muscles during intense activity when oxygen is low. Second, it is ethanol. This is produced by yeast and some bacteria in the absence of oxygen. And the third product is acetyl coenzyme A. So this is formed when oxygen is present, allowing the cell to fully extract energy from glucose. So lactic acid is formed from pyruvic acid in skeletal muscles after anaerobic conditions. So this happens when we are exercising hard and our muscles don't get enough oxygen, leading to that familiar burning sensation. Now, if we talk about ethanol, it is formed when pyruvic acid in yeast under anaerobic conditions. So it is formed by oxidation of pyruvic acid in yeast when oxygen is not available. And this is the process behind fermentation. Now, the other product that is acetyl coenzyme A, which is formed from pyruvic acid inside the mitochondria when oxygen is available. So this step is very important for the cell to enter the Krebs cycle and produce energy. So here in the equation, we can see that pyruvic acid plus coenzyme A plus NAD plus in the presence of, that is catalyzed by the enzyme pyruvate dehydrogenase in the presence of Mg2 plus, we get acetyl coenzyme A plus carbon dioxide plus NADH and H plus. So this is the reaction of oxidative decarboxylation and how acetyl coenzyme A is produced. So now let's look at the next question. The energy yield in terms of ATP is higher in aerobic respiration compared to anaerobic respiration. So why is there anaerobic respiration even in organisms that live in aerobic conditions like human beings and angiosperms? Let's look at the solution. So aerobic respiration, it is a normal process in humans, that is under usual conditions. Our cells will utilize oxygen to break down glucose and release energy. Okay. Now, however, during intense activities like heavy exercise, say our muscles need much more energy and oxygen than we are supplying by breathing. So in this case, there is too much demand of energy and we consume much more oxygen to produce that energy. So this high consumption, it results in oxygen scarcity. That is, now oxygen is less, and what happens is that the muscle cells will begin to make lactic acid by the process of anaerobic respiration to fulfill their energetic needs, that is, to fulfill the energy requirements. So similarly, in case of yeast cells and some plant tissues, they use anaerobic respiration when oxygen is low. In yeast, this process produces alcohol, that is ethanol and carbon dioxide. And the same process is used in baking and brewing, where the carbon dioxide released helps the bread rise and alcohol is used in beverages. Now let's look at the next question. Oxygen is an essential requirement for aerobic respiration, but it enters the respiratory process at the end. Discuss. So let us look at the solution. So oxygen, we know, we have studied, it is the final electron acceptor in the electron transport chain. Oxygen enters at the end of the respiratory process specifically to accept these electrons. Now, without oxygen, that is, if oxygen is not present, electrons can't flow through the ETC, which will disrupt ATP production. So here in the image, it shows electron transport chain. So here we can see that oxygen is the final electron acceptor. Now the earlier stages of respiration, that is glycolysis and Krebs cycle, they don't directly use oxygen. So these steps are like the preparation phase, where the cell breaks down glucose and gathers the energy carriers. But oxygen is not needed yet. At the end of the process, oxygen combines with hydrogen ions and electrons to form H2. And this process of oxygen accepting electrons, it helps maintain the proton gradient across the mitochondrial membrane. Now, even though oxygen is used only at the last step, it is absolutely important for entire respiratory process to be completed. So here in the image, we can see that electrons are transported through the ETC, and finally, the final electron acceptor is oxygen, which combines with hydrogen ions and electrons to form water. Now let us look at the next question. Respiration is an energy-releasing and enzymatically controlled catabolic process, which involves a stepwise oxidative breakdown of organic substances inside living cells. So in this statement about respiration, explain the meaning of number A, stepwise oxidative breakdown, and B, organic substances used as substrates. So let us look at the solution. So first, let's talk about the meaning of stepwise oxidative breakdown. So respiration involves three main steps that we have studied about: glycolysis, Krebs cycle, and the electron transport chain. So there is stepwise oxidation of organic molecules in a cell through these steps. The first step is glycolysis. This is the process where glucose is split into two smaller molecules called pyruvate, and it happens in the cytoplasm. This process produces a small amount of ATP. Next comes the Krebs cycle. This takes place in the mitochondria, the cell's energy factory. So here pyruvate from glycolysis is further broken down, releasing more energy. And next comes the electron transport chain. So electrons are passed here along a series of complexes, and this movement helps generate a large amount of ATP, which the cell uses for its activities. Now, glucose passes through series of enzymatically controlled reactions, and this is finally converted into water plus ATP plus carbon dioxide. So these are the end products of oxidative breakdown of glucose. So the cell captures the energy released in the form of ATP. Now let's talk about number B, that is organic substances used as substrates. So organic substances, they are the molecules that are commonly found in living organisms, and they are the main sources of energy for cells during respiration. Now these molecules, they are usually made of carbon atoms which are arranged in rings or long chains, with other atoms like hydrogen, oxygen, and nitrogen attached. So examples include glucose, fatty acids, amino acids, and these are the basic building blocks of life. Now these organic molecules, they are burned. So they are broken down during respiration to produce energy. So when glucose and fatty acids, they are used as the main substrates for respiration, we call this floating respiration. And this is the most common type of respiration in plants. Now, if proteins or amino acids are used as the main substrates, the process is called protoplasmic respiration. And this usually happens when the usual energy sources are not available. So students, now let us look at the next question. Comment on the statement, respiration is an energy producing process, but ATP is being used in some steps of the process. Let us look at the solution. Now, respiration as a whole is an energy yielding process. So when glucose is completely oxidized, the cell produces 38 ATP molecules. Initial steps, they however require ATP investment. That is, it is important to note that the process doesn't start with energy gain right away. The initial steps, they require the cell to invest some ATP. So the first ATP investment occurs during glycolysis. What happens here? The cell uses 2 ATP molecules to convert glucose into glucose 6-phosphate. Okay. Then another 2 ATP molecules, they are used to form fructose 1,6-bisphosphate. So in the early steps of glycolysis, a total of 4 ATP molecules are spent to prepare glucose for further breakdown. Now, why is this ATP investment necessary? So first, this ATP is used to activate glucose molecules. So by adding phosphate groups, the cell makes glucose more reactive and ready for next steps. And second, the ATP helps overcome energy barriers. Because chemical reactions, often they need a little push to get started. Third, this ATP investment drives the reactions forward. So it ensures that the process keeps moving in the right direction, leading to eventual production of more ATP. Now, the energy that is invested in these early steps, it is recovered later in the process. So during complete respiration, the cells end up with a net gain of ATP, making the initial investment worthwhile. So let's see the next question. Why is the respiratory pathway referred to as an amphibolic pathway? Let us look at the solution. So let us see why respiratory pathway is called amphibolic. This is because it serves both breaking down and building up roles in the cell. So what we mean by amphibolic pathway is that it is a pathway that involves both breakdown and synthesis reactions, that is both catabolic and anabolic reactions. So in case of catabolic reactions, molecules are broken down for energy, and in case of anabolic reactions, new molecules are synthesized. So respiratory pathway, it breaks down glucose to produce energy, and because there is breakdown, we are calling it catabolic. On the other hand, this pathway also provides intermediates that are used to build important molecules like amino acids, fatty acids and nucleotides. And therefore, this is the anabolic site where the cell is using pieces from breakdown to make new products. So here in the image, you can see amphibolic pathway. So respiratory pathway is both anabolic as well as catabolic. There is synthesis of larger molecules from smaller ones with the input of energy, as well as there is breakdown of larger molecules into smaller ones with the release of energy. And therefore, the respiratory pathway is an amphibolic pathway. Now, the citric acid cycle, it acts as a hub connecting various metabolic pathways. And it generates precursor metabolites for gluconeogenesis. So it means that it provides raw materials needed to make new glucose molecules. So this pathway is also versatile because it can utilize different substrates like carbohydrates, fats, and proteins. Now, the amphibolic pathway, it helps maintain a balance between energy production and biosynthetic processes. And therefore, respiratory pathway is referred to as amphibolic pathway. So let's look at the next question. Do you know any step in the tricarboxylic acid cycle where there is substrate level phosphorylation? So we have to tell which one step. Let's look at the solution. So let's look at the step that involves substrate level phosphorylation in the TCA cycle. So the conversion of succinyl coenzyme A to succinate is the only step in the TCA cycle with substrate level phosphorylation happening. This reaction is made possible or catalyzed by the enzyme succinyl coenzyme A synthetase, which is also known as succinate thiokinase. Now, during this step, the high energy thioester bond in succinyl coenzyme A is transformed into a phosphate bond in GTP. So here in the image, it shows citric acid cycle. So the step where succinyl coenzyme A is converted to succinate is where the substrate level phosphorylation is taking place. And it is catalyzed by the enzyme succinyl coenzyme A synthetase. Now, we see that GTP is then converted to ATP by the enzyme nucleoside diphosphate kinase. So this step ensures that the energy captured as GTP, it can be used in the same way as ATP. So this means that for every turn of the TCA cycle, one ATP equivalent is generated directly through substrate level phosphorylation. So here we can see that in this step, ATP, that is GTP or ATP equivalent, is produced per turn of the cycle. So let's see the long answer type questions. So the first question here is, in the following flow chart, replace the symbols A, B, C and D with appropriate terms and briefly explain the process and give any two applications of it. So let's look at the solution. So now if we talk about the figure, the metabolic pathway that is given is fermentation. So the products marked as A, B, C and D are as follows. Let's look at the products. So A is glyceraldehyde 3-phosphate. This is an intermediate in glycolysis. B is phosphoenol pyruvic acid. So this is the end product. This is near the end of glycolysis, important for final steps of energy release. C is ethanol, which is produced during alcoholic fermentation, especially in yeast in the absence of oxygen. D is lactic acid, which is produced during lactic acid fermentation. It occurs in some bacteria and in our muscles during intense exercise. So here in the image, we can see that glucose, it is converted to glyceraldehyde 3-phosphate, which is A, followed by formation of 3-phosphoglyceric acid, and then we have phosphoenol pyruvic acid, which is B. Then this pyruvic acid, in the absence of oxygen, there is fermentation. So we get ethanol plus carbon dioxide or lactic acid. Ethanol is C and D is lactic acid. So the fermentation we talked about is of two types. There is alcoholic fermentation in yeast. It is a classic example of how cells generate energy without oxygen. So there is incomplete oxidation of glucose under anaerobic conditions. So what happens in alcoholic fermentation? Yeast converts pyruvic acid into ethanol and carbon dioxide through two main reactions. First, in the first step, pyruvic acid, it is decarboxylated, resulting in the formation of acetaldehyde and carbon dioxide. With the help of enzyme pyruvic decarboxylase. So here in the equation you can see pyruvic acid is converted to acetaldehyde, and this reaction is catalyzed by the enzyme pyruvic decarboxylase. So this is the first step. Then in the second step, acetaldehyde is reduced to alcohol by NADH2. So here we can see acetaldehyde, it is converted to ethanol or ethyl alcohol, and there is release of carbon dioxide. Now let us talk about the second type of fermentation that takes place in muscles, that is lactic acid fermentation. So in animal tissues like muscles, when oxygen is not enough, pyruvic acid is reduced to lactic acid by the enzyme lactate dehydrogenase. So the reducing agent in this process is NADH plus H plus, which gets reoxidized to NAD plus in the subsequent processes. So this recycling of NAD plus is important because it allows glycolysis to continue. Now let us look at some applications of this fermentation process. So first is manufacture of ethyl alcohol or ethanol. So this is important for making alcoholic beverages and also for producing biofuels. And the second application is the curdling of milk to make curd. And this is aided by bacteria lactobacillus. So here in the image you can see fermentation. Incomplete oxidation of glucose gives us pyruvate. In the absence of oxygen there is fermentation. So this pyruvate or pyruvic acid is either goes through alcoholic fermentation or through lactic acid fermentation. So from lactic acid fermentation, from aspergillus and lactobacillus, we get products like soy sauce and cheese and yogurt. And in case of alcoholic fermentation, we get products like wine, sliced bread and beer from Saccharomyces. So there is production of ethanol and carbon dioxide in this case. So this was all for today's class. Thank you.